Ness. I'm from Brown University. Today I'm going to tell you a little bit about some of the work that we've been doing with barnacles. All right, so the general theme that basically centers most of the work that we do is this old but important question in the field of population genetics, which is what is the genetic basis to adaptations to ecologically heterogeneous environments. Now, for our work, we mostly utilized the Rocky Intertidal as a natural laboratory. And we pay particular attention to several dynamics of the northern acorn barnacle, or Sylvanus polonoides. Now, we utilize this little critter for a variety of reasons. There is almost a century's worth of ecological knowledge available to us, animal phylogeny, circumpolar distribution, as well as biodispersal capabilities. Now, on top of that, there is this really interesting characteristic of the barnacle, which is that it inhabits the rocky intertidal. The rocky intertidal is the space that exists basically between the high and the low tide, and is characterized by having a wide variety of abiotic and biotic stressors that can be easily tracked. Now, for today's talk, I want to pay attention to two particular axes of abiotic variation. The first one is the stress gradient that is produced by the changes in the tides, as well as the stress gradient that is produced by having different sun aspects in which barnacles ultimately settle on the shores. Now, something else that's really interesting about using this organism is that there's a long tradition of surveillance in the field of ecological genomics. So much so that we have effectively characterized genetic variation in the barnacle with every method in the book, from Alzheimer's to amplicons. Most recently, we've been doing a lot of work with next-generation sequencing. However, today I want to go old school, and I want to revisit some of the old stories from the first investigations done in ecological genomics, many of which center attention in this one gene, the mannose 6-phosphate isomerase gene, or MPI for short. Now, what's interesting about MPI? Well, it is a very important glycolytic gene that is involved in the metabolism of mannose. And mannose has a connection to the ecology of the intertidal because it is a very important sugar in the diets of many organisms that inhabit the rocky shore. I have to make a very important clarification. So throughout the remainder of my talk, I am going to mention, I'm going to refer to the genetic variation in this gene as the fast alleles of MPI and the slow alleles of MPI. This has nothing to do with any sort of kinematic properties of the protein, but rather it's a legacy labels, if you will, from the time of the Alzheimer's where this investigation started. All right. So the biology of MPI in the intertidal is rather peculiar. Most of the data that I'm going to show you, here was collected here in the United States in Maine. So these early studies really capitalize on the extreme stress gradient that is maximized in the model that I showed originally, mostly the most stressful side up here and the most benign side down there. And what they did is that they basically genotyped individuals in the most stressful edge of the intertidal and the most benign side. And what was discovered is that there's this really basically whopping sign of zonation happening at MPI. And by that, what they meant is that one of the fast, in this case, allele of MPI is really abundant in the most stressful site. There is no such observation in the most benign site, and if you genotype other alleles, like other glycolytic enzymes or the mitochondria, you do not see any observation of that phenomenon. So for almost 15 years after this was originally observed, there was a series of ecological, biochemicals, laboratory experiments that effectively created a very strong connection between fitness in the field, in the lab, and this aspect of zonation. With that data, the authors effectively proposed this idea that what's going on in the intertidal is that these two alleles may be maintained in a system of balancing selection. You may imagine if the challenges of zonation are ubiquitous, this is basically a case in which variation is maintained across the space. Now, that's a pretty elegant hypothesis, especially given the data that was collected. But when other studies attempted to gain a broader insight into this hypothesis, some interesting problems started to appear. If you repeat the experiment up in Canada, you discover that the signature of zonation in the intertidal is not apparent. Rather, zonation appears to happen at the mesohabitat level. And if you repeat the experiment here in Rhode Island, you find out there are some intertidals that have no effect of zonation, no signatures of zonation, and those that do show a reverse effect. So basically, this generated a degree of complexity for the hypothesis of balancing selection that, as you may imagine, the Allozyme technology had no chance of resolving. But now that we have some whole genome data and some panels of genome-wide variation, we can actually go back to this question and try to gain further insights. For today, I want to tell you something a little bit about the genetic basis of the MPI Alzheimer's. And I want to comment a little bit on the evidence for balancing selection, as well as this issue of reverse zonation in Maine and Rhode Island. All right. So as I mentioned, one of the things that we've been doing over the past 5 years is assembling a genome and a transcriptome. So we went into the genome, we extracted the scaffold that contains the MPI gene, shown here with 6 exons. In addition to that, we have collected data using next-generation sequencing and some old-school methods such as cloning to generate a panel of diversity of barnacle populations. And with the panel of diversity, we were able to pinpoint the mutations that appear to drive the fast and the slow allozyme variation to these 3 mutations in exon 6. So if you take that data and then do something like a phylogenetic tree, for instance, you in fact show that there is some support for the existence of 2 haplogroups. So basically, these are all the haplotypes that would produce a fast allozyme. These are all the haplotypes that would produce a slow haplotype allozyme. But what's more interesting here is that if you then group the tree with the sister taxa, say Balanced Karyosis, what you discover is that neither of these 2 mutations, or I suppose these 2 mutations are not a transpecies polymorphism, and that in fact the slow mutation appears to be a derived state in the system. All right, so this actually is a good segue to the story of balancing selection, because the main question that's always been in literature for this system is whether or not this structure is the result of a neutral process, or whether or not balancing selection may have produced the maintenance of these 2 haplogroups. So to do that, we can actually, once again, take advantage of this topology, and we can simply simulate a neutral process. So basically, what would be the amount of genetic variation that we would expect in a gene like this if the only thing driving variation was, say, a neutral coalescent process? And so the dashed line is the expected amount of variation, and the purple envelope is a confidence interval. And then what we can do is we can plot our data. Now, if you plot our data, you actually realize that there are 2 areas in the gene that appear to have an excess of variation relative to the original envelope of expectation. Now, those cases of having a very high excess of variation relative to neutrality are sort of a canonical footprint of balancing selection. However, the complexity here is that there are 2 peaks, not just the one over exon 6. Moreover, if you then conduct some more classical analysis, something like a DNDS test in this case, you do observe that the 3 mutations that we originally characterized do appear to be sort of candidates for balancing selection here, but then there are other candidates throughout the gene that add sort of additional levels of complexity for the story. All right, so there are definitely some footprints of balancing selection in the system. What about the issue of biogeographic generality? So to tackle this issue of how general are these patterns, we went back to the same places where the original analyses were conducted in Maine and Rhode Island. And what we did is that we collected hundreds of barnacles. We extracted DNA from those barnacles, and then we genotyped, or I guess we surveyed genetic variations in those populations using a method known as pooled sequencing. Now, once again, we extracted the region of the whole, and this is something that we do for the whole genome. However, we extracted the region that corresponds to MPI, and then we estimated a summary statistic known as Tejima-D. You can learn a lot of things from Tejima-D. However, under a scenario of sort of canonical balancing selection, you would hope to see an enrichment of really positive Tejima-D around the balanced polymorphism, and I hope you get an appreciation that that seems to be the case. What's interesting here is not only the fact that we see the signature of positive Tejima-D, but the fact that that is true for both the Maine population and the Rhode Island population, right? So despite the fact that there is this disagreement between the zonation stories, there is definitely some agreement in the molecular footprints that we see of the gene. All right, so what about the zonation itself? So once again, we utilized the power of pool-seq, but our sampling design for this experiment was much more granular. Instead of sampling hundreds of barnacles throughout the broad swath on the intertidal, we sample about 37 to 40 barnacles from replicates in the most stressful side and the most benign side. So again, sort of replicating the original design of the first studies. And then with that, what we conducted was an association analysis. And basically, what I'm going to show here on the x-axis are the different positions across the exons of MPI. And here is a p-value that'll give you some idea of how strong is the association of allele frequencies and dissonation. Circles will be non-synonymous mutations, and triangles will be synonymous mutations. So let's take a look at Maine. All right, so if we look at the data in Maine, the vast majority of mutations don't really show a strong enrichment or strong association between the high and the low stress microhabitats, with one exception, which is that bubble over there, which is right over exon 6, which effectively corresponds to the mutations that drive the fast and the slow state that that we sort of discovered a couple of slides ago. So that's pretty interesting because effectively we have replicated the same results that the original Allozyme papers showed, but through a completely different methodology and 20 years after the fact. That's cool. Now what about Rhode Island? All right. So once again, we have replicated the analysis that was done with Allozymes back sort of 20 years ago, and we have again shown that there is no signatures of zonation operating in our two Rhode Island samples. So this is basically the title of my talk, From Allozymes to Whole Genomes and Then Back Again, because we have effectively recapitulated the original findings of the Allozyme studies. So after all of this, what have we learned? All right. So these are the three original questions that I posed. So the first thing, in terms of the molecular basis, we have characterized those three mutations that appear to have very strong explanatory power as to whether or not they're going to be a fast or a slow allozyme. So that's new. In terms of evidence for balancing selection, I think that we have shown that there are footprints that definitely appear canonical cases of balancing selection, and that those footprints appear to be sort of consistent in both the Rhode Island and the Maine populations. But however, what appears to be clear in our data is that the zonation process remains a habitat-specific issue, which is whether or not balancing selection operates at particular spatial scales, whether a particular microhabitat will manifest the differential in fitness values that were described in the original papers is probably dependent on a collection of or interactions of ecological factors happening at the habitat level. Now the last thing, or the last thought that I want to share, is the idea that this is just one gene, as you may imagine. This is a very complex process in which many, many genes are probably responding to the challenges of intertidal zonation. And now that we have a genome, what we're working on right now is trying to characterize what other genes are involved. So hopefully next time that we see each other, I'll have that story for you. So watch this space. And of course, there's many people currently working on this and that make this research possible, as well as funding agencies. And I'll happily take questions. Yes, hi.
So what if you see a signature of balancing selection even when you don't see the donation in Rhode Island? So what do you think is the source of the balancing selection in the Rhode Island population? Or is it just bleeding over? Just being so, where is it?
Yeah, so one possibility is that the scales at which this thing manifests itself are much larger, and so populations feed into each other. A second possibility is the fact that, and there is a data set that is published that shows that there's a very strong interaction in the mortality or the of barnacles that recruit to these habitats when you account for salinity changes or salinity challenges and thermal changes. And the interaction between salinity challenges and thermal challenges is much larger than the effect of temperature or salinity alone. Now, our sites in Maine are basically in the delta of a river, which flushes constantly. Our sites in Rhode Island are basically just a regular bay, so it has a different osmotic dynamic. And that's one of the things that we believe may be related to the story, which is there's just an interaction event that is unique to some micro